Hi, good day to Prof. Wan Julian. My name is Akila binti Mokhtar with matrix number A182709, and me and my group members will present on whole genome sequencing of non-tuberculous mycobacterium or NTM isolates from the sputum specimens of cohabiting patients with NTM pulmonary disease and also NTM isolates from their environment. As an introduction, the non-tuberculous mycobacterial pulmonary disease or NTM-PD has increasing in developed countries. The development of the next generation sequencing technology helped identify the SNPs by the whole genome sequencing. The source of infection for pathogens can also be traced. The NGS studies rejecting the case that NTM-PD acquire NTM only from their environment and not from the human-to-human transmission. The 3 pairs of NTM-PD patients with no immunodeficiency and cohabiting for 15 years have also been diagnosed. For the objective of the studies is to understand the source of infection using WGS or whole genome sequencing by investigating the genomes of the NTM isolates from patients and also samples from their houses. The picture on the right side would be the whole genome sequencing process.
Methodology. The first methodology is patient enrollment. Pairs of adult patients living together and diagnosed with NTM-PD at Seoul National University Hospital satisfying the following inclusion criteria were enrolled. The second methodology is sample collection and NTM isolation. Isolated NTM from participants' sputum or bronchial wash specimens from participants was collected. Patients' homes were visited to collect environmental specimens. There are 2 types of samples, which are water samples and swab samples. As for the water samples, they were collected aseptically directly from all faucets and showers in bathrooms and kitchens. The specimen was then passed through a 0.45 micrometer filter. Then filters were rinsed and transferred to Middlebrook 7H11 plates and microbacterial growth indicator tubes (MGITs). As for the swab samples, they were taken from all the inside of the faucets and shower heads in bathrooms and kitchens. They were then washed, processed in the same manner as the water samples. All culture-positive MGITs were transferred to new Middlebrook 7H11 plates. If the culture-positive have more than 2 morphologically distinct colonies, they will be then separately transferred to 3% Ogawa media plate, incubated and purified as a single colony. If the culture is homogeneous and discrete colonies, the isolates will be collected without a further purification step.
Next is DNA preparation and sequencing. All biomass from Middlebrook 7H11 plates and Ogawa media plates are mixed with 435 to 600 micrometer glass beads and vortex for 5 minutes, incubate 80 degrees Celsius for 10 minutes, and centrifuge. Extractions of DNA from supernatant is done using QIA-AMP DNA mini kit, while DNA sequencing is done using Illumina HiSeq or NovaSeq instrument. To validate the method, 2 patient isolates and an environmental specimen isolates were sequenced twice as replicates. 16S RNA and RPOB gene sequence analysis is performed to identify each isolate from the patients. The next step is variant calling and phylogenetic analysis. Kraken-2 is used to identify reads from each isolate mapping to reference genomes. M. avian subspecies homoecious reads is mapped to M. avian subspecies homoecious TH15 using BWA, while M. intracellular reads is mapped to M. intracellular ATCC 13950. Isolates assigned by Kraken-2 as non-NTP species were excluded from further analysis. Variance calling using SAM tools and BCF tools with filling filters, which is minimum base quality of 50, minimum mapping quality of 30, support at least 4 reads and absence of heterozygosity. High confidence SMP is defined as SMPs in the core regions of each NTM species. SMP distance and the number difference of high confidence SMPs were calculated for each pair of isolates. Phylogenetic analysis is done using MegaX, while genetic distance between isolates is calculated using maximum composite likelihood method, and phylogenetic tree is constructed using neighbor joining methods.
Okay, now I will be explaining about the results obtained from this study. The first is the characteristics of the patients chosen for this study. There are 3 pairs of patients with non-tuberculous mycobacterial pulmonary disease. The first pair is a mother and a daughter with mycobacterial avian pulmonary disease who live in an apartment in an urban area for 15 years. The other 2 pair is a couple where the first couple with mycobacterial intracellular pulmonary disease who live in a house in a rural area for 30 years, and the second couple, one of whom was infected with M. intracellular PD and the other of whom was infected with Mycobacterial abscessus subspecies Macilians PD, who live in an apartment in an urban area for 30 years as well. Actually, there are other NTM species as well, such as M. kansasi and also M. xenopi. From this data, they perform a whole genome sequencing using the 8 patients' NTM isolates as well as environmental specimens. All the patients are not suspected of any immunodeficiency disorders. They are HIV negative, not taking any immunosuppressants, and have no history of recurrent infection of any organs. This has been shown because they don't want the results obtained to be due to other diseases or factors, such as If a patient is a HIV positive, this patient will be more vulnerable to other infections because they have a weakened immune system. The source of NTM for the mother and daughter and the first couple pair is from the sputum. Sputum is a mixture of mucus coughed up from the respiratory tract and the saliva. The sources of NTM for the other pair of couples is from the bronchial washing. Bronchial washing is where a mild salt solution will be used to wash over the airway surface to collect the cells, and then it will be observed under a microscope.
The second result is on NTM isolation and sequencing. On average, 19.8 million sequencing reads were obtained for each isolate. According to Kramer-based taxonomic classifications of NGS reads, isolates from patient A and patient B were identified as Mycobacterium avium subspecies hominis. Isolates from patient C, D, and E were identified as Mycobacterium intracellular. One isolate from patient F was identified as Mycobacterium abscessus. Two isolates from environmental specimens in home 1 were identified as Mycobacterium fortituum. The remaining 4 isolates from home 1 and 3 isolates from home 3 were classified as non-NTM and were also excluded. Based on the distributions of SNP allele fractions, all isolates were monoclonal except for the isolate from patient C, which consisted of 2 clones at an 8:2 ratio. The 25,441 and 2,485 high confidence SNPs from patient C were belonging to the C major and C minor clones based on SNP allele fraction. The 3 pictures at the side are an example of how those Mycobacterium species look under a microscope.
As for pairwise SNP distance and phylogenetic analysis, this slide shows the pairwise SNP distance. The first is the SNP distance between patient A and patient B, which is 14,768. Next is the SNP distance between 3 residues, which are the isolates of patient A, kitchen sink faucet 1 and kitchen sink cold water 3, which is less than 100. As for SNP distance between patient C major with patient D and patient C minor with patient D are 29,873 and 12,670 respectively. They are higher than those compared to patient E. From this slide, it shows the phylogenetic analysis. From figure A, the first one shows that the isolate from patient A and its replicate cluster with the specimens from the kitchen, and the second one shows that the isolate from patient B cluster with the isolates from bathroom and kitchen. From figure B, we can see that phylogenetic analysis have confirmed that all 3 isolates from patient C, D, and E were not closely related to each other.
Next, we move on to the discussions. No pairs of patients had near identical of NTM isolates. Figure 2 from the next slide shows the evidence of the strains. Second one, the one patient pair are infected with Mycobacterium intracellular and another pair got infected by another different species of NTM. So this shows that patient-to-patient transmissions is very less likely to happen. The third discussion would be the patient isolate and plumbing isolate have same repetitive sequence based on PCR DNA fingerprint. This is because of the major reservoir of the Mycobacterium are from household water. Next, based on the studies, 35% patients with NTM pulmonary disease had NTM isolates that are similar to the ones from the household water. Next, the whole genome sequencing revealed that the 52% patients with Mycobacterium avium pulmonary disease genetically matched to the household isolates too. Lastly, a phylogenetic tree based on the whole genome sequencing consistently reports that the source of Mycobacterium avium pulmonary disease patients are from the kitchen and the bathroom.
Now, I will continue the discussion for this study. From the 50 water samples, each from home 2 and home 3, they could not culture any NTMs from home 2 and only identified 1 NTM from home 3. And this may be due to other sources of NTM-PD other than water. NTM, which is an environmental organism, can be found in soil and dust. Patients from home 2 were farmers and their home was in a high soil environment. So source of M intracellular is soil instead of water. Since I mentioned earlier that NTM can also be found in soil. Also, all the isolates from home 3 were 3 morphologically distinct colonies were cultured from 1 environmental specimen were not NTM. This can be proven by the sequencing risk of NTM isolates from patients E and F aligned to NTM reference genome, while those from environmental specimens isolates do not align. Therefore, they concluded that environmental isolates are not closely related to NTM in home 3. From this study, patient E, who has a M intracellular PD in home 3, has been exposed to NTM outside the house. According to Thompson et al., there are factors that cause difficulties in identifying the source of NTM, such as the lag time between the acquisition of NTM from the environment and diagnosis of NTM PD. In many cases, a patient had symptoms for several years before being diagnosed, making it challenging to pinpoint the environment to which they were exposed. Patients may also encounter NTM outside the home.
Lastly, Based on these studies and research, we can conclude that the three pairs of cohabiting patients determined to have different strains according to the whole genome sequencing data. Therefore, the likelihood is minimal for the patient-to-patient transmission. Thank you. That is all from us.